In this video, we're going to be covering culturing microorganisms, and this is our learning objective, to be able to describe how to culture microorganisms using aseptic techniques. As always, all the timestamps will be in the description below, so feel free to skip to whatever part you need help with. These are our three definitions for this video. The first one is nutrient broth, which is a liquid medium that contains nutrients required for the growth of microorganisms. This includes things like carbohydrates and minerals. Second one is a petri dish, which is a shallow, circular, transparent dish with a flat lid, which is used to culture microorganisms. The third is agar jelly, which is a jelly-like substance placed in our petri dishes, and that is also used to help culture microorganisms. For this video, you should have a good knowledge of why we actually need to culture microorganisms in the first place in order to study them. And the basics of that is that they're so small that in order to be able to study properly the processes which occur within them, we need to have a large amount of the bacteria, for example, in quite a small area of space. So we might have thousands of bacteria on a small petri dish, so all their processes are amplified in that one small space. All the information in this video also relates to a required practical, which is on your spec, which is basically the same thing, culturing microorganisms using these techniques. And this video will tell you why each step in that experiment is important and how that relates to the biology of bacteria. To start off, these are the main two ways within which we culture microorganisms in a lab. So the first one on the left is a nutrient broth, and this is essentially a liquid medium in which we have our bacteria that we're growing, and we also have a lot of the nutrients required for the growth of the bacteria. So this could include things like carbohydrates, so for example glucose, which bacteria can use for respiration in order to produce energy for replication, and then also amino acids, which are obviously really important in protein synthesis. And then generally you might have minerals as well as other factors which are important in bacterial growth and replication. Then on the right we have an agar gel plate. So this consists of our petri dish, which is our flat-bottomed dish, which also has a lid, which we can shut, and we can make sure that there's no contamination of our bacteria once we've put it onto our plate. And we also have some agar jelly, which is hot and sterilized when it's poured into our petri dish, and then it will solidify, and it basically provides a platform for our bacteria to grow. In order to transfer or to grow bacteria on our agar gel plate, we need an inoculating loop, which is essentially a loop of wire which we can dip in our nutrient broth. We can spread it over our agar gel plate to make our bacterial culture. Now we're going to move on to the aseptic techniques that we use to culture microorganisms in order to prevent contamination. So the first one is that petri dishes and all of our culture media, so that includes things like our inoculating loop and the head or the neck of any bottles that we use, they're all sterilized. And this prevents introducing other foreign bacteria into our sample. And if this happens, it could be harmless, but they could also compete with the bacteria that we're studying. They could use up all the nutrients, so at the end we don't get the growth of the bacteria that we want. And we could also introduce harmful bacteria which could lead to harm to ourselves or to our classmates. So similar techniques to avoid contamination are making sure that the lid of the petri dish is sealed for the same reasons, and also we tend to store our petri dishes upside down, and this helps stop airborne microorganisms from contaminating the culture, and the kind of storing it upside down ensures that condensation doesn't land on our surface of our agar jelly and disrupt the growth of our bacteria. However, an important point with sealing our dish is that it shouldn't be sealed all the way around, so no oxygen can get in at all, because obviously bacteria need to respire and that requires oxygen so they can grow. So we do want some oxygen to come in, and if we completely cut off all the oxygen from our sample, that could encourage the growth of harmful anaerobic bacteria which don't need oxygen to grow. Finally, we incubate our culture at 25 degrees, and this is kind of an optimum temperature which allows rapid bacterial growth. But also, if we did it at a higher temperature, which is closer to our internal human temperature, so for example 37 degrees, it would be more likely that bacteria which are harmful to human health would grow, and obviously we don't want that at all. And then in the picture below you can kind of see aseptic techniques in action. So the person is spreading the bacteria on the agar jelly, but they've only got the lid open kind of at a 45-degree angle, just again to minimise the risk of contamination by airborne microorganisms. If the microorganisms that we're culturing are bacteria, we can use them to test the effectiveness of different antibiotics. This involves obtaining a small paper disc or filter paper which is soaked in a particular antibiotic, and then placing it on our bacterial culture. And if we incubate that at 25 degrees for a period of time, for example 2 days, we should get a clear zone surrounding where we've placed our antibiotic, and this shows where bacteria have been killed. So for example, in the diagram on the right, where the arrows sort of extend out, that is where bacteria has been killed, and that is known as a zone of inhibition. And in order to carry this out as an experiment, we first need a control. So we'd have a filter paper disc just with water on it, and that shouldn't obviously show any zone of inhibition or any bacterial death. However, in order to compare the effectiveness of different antibiotics, we need to measure the size of the clear area or the zone of inhibition surrounding where we placed it. And to do that, we calculate the area of the kind of circle. So we use the equation pi r squared, where r is the radius of the circle. So the radius is half the width or half the diameter. So for an example, it's the kind of pink purple line on the diagram. So the larger the zone of inhibition, the more effective the antibiotic is. This is our first worked example question for culturing microorganisms. A disc containing penicillin made a clear zone 80 millimeters across on a bacterial plate. What is the area of the clear zone? So in questions where you're not given a diagram, I always find it helpful to draw a quick one out just so I can understand what the question is asking me. So for this question, let's say our clear zone is roughly going to be a circle, and it tells me that it is 18 millimeters across, so that's the diameter. So from edge to edge, that is 18 millimeters. And as we've previously mentioned, the formula that we use to work out the area of the clear zone is pi r squared, where r is the radius. And we know that the radius is half of the diameter, so in order to find that value, all we have to do is divide 18 by 2, which is 9. And then we can pop our value into our equation to work out the area of the clear zone. So we get pi, which is roughly 3.14, but use your calculator, times 9 to the power of 2. And to two significant figures, that is 250 millimeters squared. I'm going to have to put the squared because obviously it's a value of area. And in terms of significant figures, you should normally write it to however many there are in the question. So the only value given in the question is 18 millimeters, which is obviously 2 sig fig. So that's how we're going to write our answer. So this is our second worked example question for culturing microorganisms. So feel free to pause the screen for a moment and walk through it yourself, and then I will go through it. So a student is given a tube containing a liquid nutrient medium. The medium contains one type of bacterium. Describe how the student should prepare an uncontaminated culture of the bacterium in the petri dish for 6 marks. So this question is essentially asking us about all the range of aseptic techniques that we can use to culture an uncontaminated sample. So for this question, I find it useful to break it up into 3 parts. So before inoculation, and inoculation being a transfer of the bacteria to our culture medium, during the inoculation and after the inoculation. So if we start off with before, the first really important thing is to make sure that all our culture media are sterilised. So this is things like the petri dish and our inoculating loop. So we're going to sterilise that to kill unwanted bacteria. And you should also put down how we do this. So a really kind of major way is using an autoclave, which essentially heats our materials to a really high temperature, which kills all the possible bacteria. And in terms of the loop, we can pass it through a flame to sterilise it also. So pass through flame. And then we're going to think about procedures we can do during the actual inoculation itself. So we'll put during. So the first thing is to actually transfer our bacteria to our culture medium, and we're going to do this with our inoculating loop. So we're going to spread bacteria onto our agar. And then also thinking about how we can minimise contamination during that period is to keep the lid of our dish kind of as close to the dish as possible, so only opening it at a 45-degree angle, for example. So keep lid close to dish, and that is going to prevent airborne microorganisms from entering. So next we're going to focus on how we can avoid contamination post-inoculation. So this is going to be things to do with our petri dish. So first of all, we're going to seal it with tape, but obviously not all the way around, so we don't get our harmful anaerobic bacteria. Not all the way around. And then kind of put the reason why we're doing that, so to prevent bacteria entering. And finally, we're going to incubate our sample at 25 degrees. And we pick this temperature, first of all, so that our bacteria can grow. And then also, obviously, to avoid more harmful bacteria from growing at a higher temperature. And with all of those points, you can get, well over 6 marks, 6 points there, and then pick which ones you can remember and kind of explain why you're doing them as well. So to summarise, in this video we've learnt how to culture microorganisms using a range of aseptic techniques, and also learnt why these techniques can prevent contamination. So the next step that I would advise you to take is to go through the video on the required practical attached to these techniques, just so you can put your understanding of them into practice, and then also do some questions involving calculating the clear zone or the area of inhibition using antibiotics and bacteria.